Okay, so last time we stopped with going over our complete dominance and we started to do some Punnett squares. So this time I'm going to give you guys how we can kind of go deeper into our Punnett squares and actually solve problems that might extend more than just making a Punnett square. So two different types of ratios here. We have genotypic and phenotypic. Now, let's just review genotypes and phenotypes before we get into the ratios. The genotype is going to be our combination of alleles that you have in your chromosomes. So you get one allele from mom and you get one allele from dad. When those two alleles come together, they make up your genotypes. So we had three different genotypes we went over. I used B here as an example. Big B, that's homozygous dominant, so that's one genotype. Our middle one, we had one of each allele. We had a dominant and a recessive one. We called that heterozygous. And lastly, we had our little b, which was our homozygous recessive. Those are the three genotypes we can have. It doesn't matter what letter you use. Like I said, guys, in the previous video, I really recommend when you're even doing these Punnett squares on your own, I recommend using letters where the capital and the lowercase don't look the same. Like the letter C, capital and lowercase look the same, they're just a different size. I like using B's, R's, A's, where the lowercase does not look anything like the capital. So that's the genotypes. The phenotypes, that was going to be what we see based on the genotype. So I know we did purple flowers and white flowers, and purple was dominant to white. If it was big B for our genotype, we would have purple flowers. If it was big B, we would have purple flowers as well. But if it was little b, we would have that recessive trait, which little b would be our only one that is white. And both of these would be purple. Okay. These here are our phenotypes. Okay. So what we're going to do is we're going to make genotypic and phenotypic ratios. And all this is when we do our monohybrid cross Punnett square, monohybrid meaning we're only doing one trait, we get 4 boxes. 1, 2, 3, and 4. We are just simply doing a ratio of what is in our boxes. And it could be the genotype, which is what we would exactly see in the boxes, or it could be the phenotype, which is not in the boxes, but we can draw a conclusion based on our genotype. Okay, so let's go back to the example we did last time with our Punnett square here, where we did a homozygous dominant, I think it was a purple flower, and we mixed that with a homozygous recessive one. So if we look in the boxes, let's do our genotypic ratio first. We're going to start off with big R. Well, if you look in the boxes, we're just looking in the boxes, there's none. So we're going to have 0 big R. All 4 of them are heterozygous, so we're going to have 4 big R, and we're going to have 0 little r. And I showed you all of the different genotypes. Even if we had 0, I put them in there. Now, you might see them put in, you might see them not put in. Most of the time, guys, you just have to write what you see. So in reality, I could have just write 4 big R and been done. All right. Phenotypic ratio. Now, we have 3 different genotypes, but we only have 2 phenotypes. We have purple and white. So for these ones, big R, that's going to be purple. So we're going to have 4 purple and 0 white. Okay. Let's do one more example before we move on. Let's say we cross, let's just use polydactyly as an example. So having more than 5 fingers. So it's kind of funny, but more than 5 fingers is dominant. And we will cross 2 heterozygous individuals. All right, so I'm just going to use R. Make my Punnett square here. And we're going to have big R, big R, big R, little r, big R, little r, and little r, little r. So let's do our genotypic ratio first. So if we look in there, and we're only looking at the ones inside the boxes, guys, I'm going to have 1 big R, big R. I have 2 big R, little r. And I have 1 little r, little r. Phenotypic. I'm just going to say 6 fingers. Okay. That's usually how polydactyly goes. Usually you have 6 fingers. So if you look here, we have 1, 2, 3. That would show that dominant phenotype. So we would have 3 with 6 fingers to 1 with 5 fingers. Okay? So there was just another way we can do it. Now, so far, guys, with all of our examples, we have been going over complete dominance. And what complete dominance means, and I'll write it out for you here. Complete dominance is going to be when our, sorry if you can hear the bell in the background. So complete dominance. This is when our dominant allele is completely dominant over our recessive allele. Let me show you what I mean by that. Whatever big R is, we are going to have that dominant phenotype. Whatever little r is, we're going to have that recessive phenotype. However, the heterozygous, the big R, little r, this one, we are also going to have that dominant phenotype. Okay. When we talk about complete dominance, what we're saying is that big allele, okay, the dominant allele, the big R, is completely dominant over the little r, the recessive allele. Since that is the case, we have the heterozygous phenotype showing the dominant, heterozygous genotype showing the dominant phenotype. Okay? So now we're going to go over different forms of inheritance. Now, every single trait in your bodies can be inherited. Come on, lights. All right. Every single trait in your body can be inherited differently. So one thing might be inherited by complete dominance, kind of like polydactyly, but other things could be inherited a different way, maybe incomplete dominance, and I'll show you what that means. So the first one is incomplete dominance. Now, students miss this every single year. You have to make sure that when we're talking about incomplete dominance, only our heterozygous phenotype changes. Let me repeat that in case you didn't hear it. In incomplete dominance, only our heterozygous genotype changes. Only the heterozygous genotype changes. The homozygous dominant and the homozygous recessive, the phenotypes do not change. Okay? So here's an example of this. Let's say that in flowers, certain flower, whatever it might be, let's say that blue flowers are dominant to yellow. Homozygous dominant is still going to be blue, just like complete dominance. Nothing changes there. Little b is still going to be yellow. Again, nothing changes there. However, big B, little b, our heterozygous genotype, is no longer blue. If this was complete dominance, it would be blue. But in incomplete dominance, what the heterozygous is it's a blend. It's a blend of our dominant allele and our recessive allele. In other words, guys, the one allele is not completely dominant over the other. It doesn't completely mask the other one like it did in the heterozygous. So what happens here is they blend. When we mix blue and yellow together, it makes green. We could also do this with blue and red. If we mix those together, it would make purple. All right. White and red makes pink. Right? It's different. Going over Art 101 with you guys. All right. Black wool sheep are dominant to white wool sheep. The color of the wool is inherited by incomplete dominance. So here, now that we're adding things on, guys, here's your first step all the time. All the time. Determine the type of inheritance. Is it complete dominance? Is it incomplete dominance? We're going to go over another one called CoDominance. After that, we're going to go over another one called sex-linked inheritance. Out of those four, you got to determine which one it is. And that'll be able to tell you how many different phenotypes you're going to have. If you notice, for incomplete dominance, we now have three phenotypes. We have the dominant phenotype, we have the recessive phenotype, and we have the blended phenotype for the heterozygous individual. So, what would be the color of the heterozygous offspring of the black and white sheep? So we got a black sheep, white sheep, they mate, and we get a gray sheep. All right. So, again, black is dominant, so black would be big B, white would be little b. If they mate, all the individuals should be big B, little b, meaning they would all be gray. Red flowers are dominant to white. It is inherited by incomplete dominance. What would be the genotypic and phenotypic ratio of the offspring of a flower that is homozygous recessive with a flower that is heterozygous? So let's set up our Punnett square here. We have, once again, homozygous recessive with a heterozygous individual. So homozygous recessive is our first one. We know that's going to be, and I'm just using R. You can use whatever letter you want. It doesn't matter. That one can be white, right? Heterozygous, that's going to be big R, little r. And when we blend red and white together, it's going to be pink. So let's set up our Punnett square. I'm just going to bring these down, bring these over, do your Punnett square, carry all the letters over. Now, if we want to do the genotypic ratio, I don't see any in there that are big R, so that's going to be 0. We are going to have 2 that are big R, little r. Those are the 2 on top. We're going to have 2 that are big R, little r. I'm sorry. We're going to have 2 that are little r. That's our genotypic ratio. Can also do the phenotypic ratio. So we have 3 different phenotypes for incomplete dominance. We have red, we have pink, and we have white. So in the boxes, you could even use your genotypic ratio as long as you did that correctly. 0 are big R. That means we're going to have 0 red. 2 of them are big R, little r, meaning we are going to have 2 pink. We're also going to have 2 little r, which means we're going to have 2 white. Okay? So you guys can see the difference between complete dominance and incomplete dominance, given that complete dominance we have 2 phenotypes. Incomplete dominance we have 3 phenotypes because of that blend of the heterozygous. Here's another example. So we got red, dominant to white once again, incomplete dominance once again. What would be the genotypic and phenotypic ratio of a homozygous recessive with a homozygous dominant flower? So homozygous recessive, white, homozygous dominant, big, red, do your cross. And if you look here, we got all heterozygous individuals. So if this particular flower, which every flower is going to be different, orchids are crazy. You know, if you guys have ever seen orchids, they're very pretty flowers, but they have spots all over them, some of them. Some of them are solid colors, but a lot of them do have spots, and we'll talk about that in a little bit. But every type of flower is going to inherit their petal color a little bit differently. So our genotypic ratio for this, not going to have any big R. All 4 of them are big R, little r. None of them are little r. So that's going to give our phenotypic ratio of 0 red to 4 pink to 0 white. Okay, so we'll do codominance real quick, guys, and then we will call it a day after codominance. So this is going to be our other form of inheritance. I know it says a 2 there. We could treat this as our third, given that complete dominance was first. So, and I'll make a chart for you guys after this. But we have complete dominance, incomplete dominance, and codominance. So these are our 3 right now. Once again, for codominance, and do not mistake this because students always mistake it, it's only the heterozygous genotype that is changing. Only the heterozygous. So, both alleles are going to be expressed in the heterozygous individual. What we're going to do in here, guys, and you will see it also with incomplete dominance sometimes, depending on how they do the problem, you might see 2 different letters for alleles because no allele is actually dominant. We see both of them. So they are co means together, the prefix. So together they are dominant. There is no recessive allele. So what is the genotypic and phenotypic ratio for the following? Blue flowers are dominant to red. And guys, again, every different species of flower is going to inherit their color differently. So it just depends on the species. Blue flowers are dominant to red. Flower color is inherited by codominance. Cross 2 heterozygous flowers. So let's cross 2 heterozygous ones here. Doesn't really matter what letter we use. I'm just going to use B for blue. And our other color was red. So I'm going to use R for red. Now, in the past, we have used big B, and then red would have been little b. But this is codominance. And you could still use little b if you want to. You'll still kind of get the same answer, especially for the phenotypic ratio. But I just don't want you guys to see, oh my goodness, we got 2 capital letters here and get all confused on the keystone. We don't want that. So we are going to cross, let me just make sure I got it right, 2 heterozygous flowers. So we're going to cross big B with an R and a big B with an R. All right, so we've got big B, big B, big BR, big BR, and RR. So genotypic ratio, we're going to have 1 big B to 2 big BR to 1 RR. And once again, guys, I know I keep saying this, it doesn't matter what letters you use. You know, if you wanted to use A and B, you could use that too. It doesn't matter. Phenotypic. There is going to be 1 big B is blue. 2 are big BR. Which is going to be purple. And then one of them is going to be RR. I sound like a pirate there, but that color was red. All right. Okay. So we'll do more examples with this next time, guys. I'll give you one more example with codominance, and then I'll make you a nice chart. I think I gave you enough to chew on today. I'll give you a nice chart comparing codominance, incomplete dominance, and codominance. So we'll compare all 3 of them, just so you guys can see a difference between the different genotypes on them. All right, guys, have a good rest of your day, and I'll see you next time.